The sacral plexus, formed primarily by the lumbosacral trunk and the ventral rami of the first four sacral nerves, produces the sciatic nerve, the largest nerve in the human body. This massive nerve, actually consisting of two nerves bound together by connective tissue, supplies most of the muscles of the posterior thigh and all muscles below the knee. The tibial division of the sciatic nerve innervates the calf muscles and the intrinsic muscles of the foot, while the common fibular division supplies the muscles that dorsiflex the foot and extend the toes. The organization of peripheral nerves reflects both the segmental heritage of vertebrate development and the functional demands of limb innervation. Individual muscles typically receive innervation from nerves containing fibers from multiple spinal segments, providing redundancy that preserves some function even when single roots are damaged. Conversely, individual spinal segments contribute to multiple peripheral nerves, ensuring that segmental lesions produce complex patterns of deficit rather than complete loss of function in discrete anatomical regions. The autonomic nervous system components within the peripheral nervous system follow different organizational principles than the somatic nerves. Sympathetic nerve fibers exit the spinal cord through the ventral roots of thoracic and upper lumbar segments. Then synapse in the sympathetic chain ganglia that run alongside the vertebral column. These ganglia form an interconnected network that allows for coordinated sympathetic responses across multiple body regions. The sympathetic chain extends from the base of the skull to the coccyx, with ganglia at most segmental levels connected by longitudinal nerve bundles. Preganglionic sympathetic fibers may synapse in ganglia at the same level they exit the spinal cord, travel up or down the chain to synapse at different levels, or pass through the chain ganglia to reach more distant collateral ganglia. This organization allows for both localized and widespread sympathetic responses depending on physiological demands. Parasympathetic outflow occurs through specific cranial nerves and the sacral spinal nerves, with parasympathetic ganglia typically located near or within the target organs they innervate. This anatomical arrangement allows for more localized parasympathetic control compared to the more diffuse sympathetic responses. The vagus nerve provides the major parasympathetic innervation to thoracic and abdominal organs, while pelvic parasympathetic nerves from the sacral spinal cord control bladder, bowel, and reproductive organ functions. The enteric nervous system, sometimes called the body's second brain, consists of extensive networks of neurons within the walls of the digestive tract. This system contains more neurons than the spinal cord, and can function independently of central nervous system control, though it normally receives modulatory input from both sympathetic and parasympathetic sources. The enteric nervous system coordinates the complex patterns of muscle contraction and secretion required for effective digestion and absorption. Understanding peripheral nervous system organization provides the foundation for comprehending numerous clinical conditions affecting sensation, movement, and autonomic function. Peripheral nerve injuries produce characteristic patterns of deficit that reflect the anatomical pathways and functional organization of specific nerves, while systemic conditions such as diabetes can affect peripheral nerves in patterns that reflect their metabolic vulnerabilities and anatomical distributions. The autonomic nervous system operates as the body's unconscious controller, managing the vital functions that sustain life without requiring conscious awareness or voluntary effort. This remarkable system regulates heart rate, blood pressure, breathing patterns, digestive processes, and countless other physiological functions through two complementary divisions that work in dynamic balance to maintain homeostasis and respond to changing internal and external demands. The sympathetic division of the autonomic nervous system prepares the body for action during times of stress, danger, or increased activity, often described as the fight or flight system. This network mobilizes energy resources, heightens alertness, and primes the body for rapid response to challenging situations. The anatomical organization of the sympathetic system reflects its role in producing coordinated, body-wide responses to urgent circumstances. Sympathetic preganglionic neurons originate from the thoracolumbar region of the spinal cord, specifically from the lateral horns of spinal segments T1 through L2. This thoracolumbar outflow creates a concentrated source of sympathetic activity that can be activated rapidly when circumstances demand immediate physiological adjustments. The preganglionic axons exit the spinal cord through the ventral roots along with motor neurons, but quickly separate to enter the sympathetic chain ganglia through structures called white communicating rami. The sympathetic chain ganglia form two parallel columns that extend along either side of the vertebral column from the base of the skull to the coccyx. These interconnected ganglia, numbering approximately 22 to 24 on each side, serve as relay stations where preganglionic neurons synapse with postganglionic neurons. The chain organization allows preganglionic fibers to influence multiple ganglia, enabling the coordinated responses characteristic of sympathetic activation. Preganglionic sympathetic fibers may follow several pathways after entering the sympathetic chain. Some synapse immediately in the ganglion at the same level they entered, while others travel up or down the chain to synapse in ganglia at different segmental levels. This arrangement allows thoracic preganglionic neurons to influence sympathetic outflow to the head and neck, while lumbar preganglionic neurons can affect sympathetic activity in the lower extremities. Still other preganglionic fibers pass through the chain ganglia without synapsing, continuing as splanchnic nerves to reach collateral ganglia located closer to their target organs. The collateral ganglia, including the celiac, superior mesenteric, and inferior mesenteric ganglia, provide sympathetic innervation to abdominal and pelvic organs. These ganglia receive preganglionic input through the splanchnic nerves and send postganglionic fibers to the digestive tract, kidneys, reproductive organs, and other visceral structures. The positioning of these ganglia near their target organs allows for more refined control of sympathetic activity in specific organ systems. Sympathetic activation produces characteristic physiological responses that prepare the body for increased activity and stress management. The heart rate increases and contractile force strengthens, pumping more blood to active tissues. Blood vessels in skeletal muscles dilate to increase blood flow, while vessels in the digestive tract and skin constrict to redirect blood toward more critical organs. The pupils dilate to improve visual acuity, while bronchioles in the lungs dilate to increase airflow and oxygen delivery. The adrenal medulla represents a specialized component of the sympathetic system where preganglionic neurons synapse directly with modified postganglionic cells that function as endocrine glands. These chromaffin cells release epinephrine and norepinephrine directly into the bloodstream, creating a hormonal amplification of sympathetic nervous system effects. This arrangement allows sympathetic activation to produce both immediate neural effects and longer-lasting hormonal influences that sustain physiological changes over extended periods. The parasympathetic division of the autonomic nervous system promotes rest, recovery, and digestive activities during periods of calm and safety, often called the rest and digest system. This division conserves energy, slows heart rate, stimulates digestive processes, and supports the maintenance activities essential for long-term health and survival. The parasympathetic system's anatomical organization differs significantly from the sympathetic system, reflecting its role in producing more localized and organ-specific responses. Parasympathetic outflow originates from two distinct regions of the central nervous system, creating the craniosacral pattern that characterizes this division. The cranial component arises from nuclei within the brainstem and travels through four cranial nerves: the oculomotor, facial, glossopharyngeal, and vagus nerves. The sacral component originates from the lateral horns of spinal segments, S2 through S4, and forms the pelvic nerves that innervate pelvic and lower abdominal organs. The vagus nerve carries the majority of parasympathetic outflow, providing extensive innervation to thoracic and abdominal organs. This remarkable nerve, the longest of the cranial nerves, sends branches to the heart, lungs, stomach, liver, pancreas, and most of the small and large intestines. Vagal stimulation slows heart rate, increases digestive secretions, stimulates intestinal motility, and promotes the restorative processes that occur during rest and feeding. Parasympathetic ganglia typically lie near or within the organs they innervate, creating short postganglionic fibers that provide precise local control. This anatomical arrangement contrasts sharply with the sympathetic system's long postganglionic fibers and allows parasympathetic activity to be more selective and organ-specific. The positioning of parasympathetic ganglia also provides some protection from systemic influences that might otherwise interfere with local regulatory processes. The integration of sympathetic and parasympathetic activity creates the dynamic balance essential for optimal physiological function. Rather than operating as simple on-off switches, these systems provide graded responses that can be fine-tuned based on immediate needs and long-term regulatory requirements. The balance between sympathetic and parasympathetic tone varies throughout the day, with sympathetic activity generally higher during waking hours and parasympathetic activity dominating during sleep and rest periods. Autonomic reflexes provide rapid, unconscious adjustments to physiological challenges without requiring higher brain involvement. The baroreceptor reflex exemplifies these automatic responses, using pressure sensors in major arteries to detect changes in blood pressure and triggering appropriate cardiovascular adjustments. When blood pressure rises, increased baroreceptor firing inhibits sympathetic activity and stimulates parasympathetic output, slowing heart rate and reducing vascular tone to lower pressure back toward normal levels. Hypothalamic control centers integrate autonomic function with endocrine responses and behavioral patterns, creating coordinated physiological programs appropriate for different circumstances. The posterior hypothalamus contains sympathetic control centers that can trigger fight or flight responses, while anterior hypothalamic regions house parasympathetic centers that promote rest and recovery. These hypothalamic regions receive input from throughout the nervous system, allowing emotional states, circadian rhythms, and conscious intentions to influence autonomic function. Circadian rhythms impose daily patterns on autonomic function that prepare the body for the predictable demands of waking and sleeping. Sympathetic activity typically peaks during late morning and early afternoon hours, supporting the increased metabolic and cardiovascular demands of daytime activities. Parasympathetic activity becomes more prominent during evening and nighttime hours, supporting digestive processes, tissue repair, and the restorative functions of sleep. Stress responses demonstrate the autonomic nervous system's capacity for rapid, comprehensive physiological adjustments. Acute stress triggers immediate sympathetic activation that mobilizes energy stores, increases cardiovascular output, and heightens alertness and sensory sensitivity. Chronic stress can lead to sustained sympathetic activation that may contribute to cardiovascular disease, digestive disorders, and immune system dysfunction, highlighting the importance of maintaining appropriate autonomic balance. The autonomic nervous system's influence extends far beyond the traditional boundaries of involuntary function, affecting immune responses, hormonal secretions, and even cognitive processes. Recent research has revealed extensive connections between autonomic centers and brain regions involved in emotion, memory, and decision-making, suggesting that autonomic function both influences and is influenced by psychological states and conscious experiences. Understanding autonomic nervous system function provides insight into numerous medical conditions and therapeutic approaches. Autonomic dysfunction can result from diabetes, neurodegenerative diseases, or medications producing symptoms ranging from blood pressure instability to digestive disorders. Conversely, techniques that influence autonomic function such as deep breathing exercises, meditation, and biofeedback can provide therapeutic benefits for stress-related conditions and support overall health and well-being. Sensory pathways represent the neural highways that transform physical stimuli from the environment and within the body into the conscious experiences of touch, temperature, pain, and spatial awareness. These sophisticated information processing systems begin at specialized receptors distributed throughout the body and culminate in specific regions of the cerebral cortex, where raw sensory data becomes integrated into meaningful perceptions that guide behavior and inform our understanding of the world around us. The somatosensory system processes information about touch, pressure, vibration, temperature, pain, and body position through two major ascending pathways that differ in their anatomical organization and functional specialization. These pathways demonstrate the nervous system's elegant solution to the challenge of transmitting different types of sensory information with the speed and precision required for survival and complex behavior. The dorsal column pathway carries information about fine discriminative touch, vibration, and proprioception from mechanoreceptors located in skin, joints, and muscles throughout the body. This pathway maintains exceptional spatial resolution and temporal precision, allowing humans to perform delicate manipulative tasks and maintain accurate awareness of body position in space. Primary sensory neurons with large, heavily myelinated axons carry these signals at velocities approaching 100 meters per second, ensuring rapid transmission of information critical for motor control and spatial navigation. The organization of the dorsal column pathway reflects the principle of somatotopic mapping, where adjacent body regions are represented by adjacent neural pathways throughout the entire system. Primary sensory neurons enter the spinal cord through dorsal roots and ascend ipsilaterally in the dorsal columns, with fibers from the legs and lower trunk traveling in the fasciculus gracilis and fibers from the arms and upper trunk ascending in the fasciculus cuneatus. This segregation maintains the orderly topographic representation that characterizes somatosensory processing. The dorsal column fibers terminate in the nucleus gracilis and nucleus cuneatus within the medulla, where they synapse with second-order neurons that give rise to the medial lemniscus. At this point, the pathway crosses the midline at the internal arcuate fibers, creating the decussation that results in each cerebral hemisphere processing sensory information from the opposite side of the body. The crossed fibers ascend or is the brainstem as the medial lemniscus, maintaining their somatotopic organization, while projecting to the ventral posterior nucleus of the thalamus. The spinothalamic tract processes information about pain, temperature, and crude touch through a different anatomical route that reflects the urgent nature of these potentially life-threatening stimuli. Primary sensory neurons, with smaller, less heavily myelinated or unmyelinated axons, carry these signals more slowly but often, with greater emotional and motivational significance than the mechanosensory information processed by the dorsal columns. Unlike the dorsal column pathway, spinothalamic tract fibers cross the midline shortly after entering the spinal cord, within 1 or 2 segments of their entry level. This early decussation means that unilateral spinal cord damage can produce different patterns of sensory loss on the 2 sides of the body, with fine touch and proprioception lost ipsilaterally below the lesion level, while pain and temperature sensation are lost contralaterally. The spinothalamic tract ascends through the lateral white matter of the spinal cord and continues through the brainstem to reach the ventral posterior nucleus of the thalamus. Along its course, the tract gives off collateral branches to brainstem reticular formation nuclei that contribute to arousal and attention mechanisms, ensuring that painful or potentially threatening stimuli can alert the organism and mobilize appropriate behavioral responses. Trigeminal sensory pathways process somatosensory information from the face and head through anatomical routes that parallel those used for body sensation. The trigeminal nerve's 3 divisions collect sensory information from the forehead and scalp, the cheek and upper lip, and the jaw and lower lip respectively, maintaining a precise topographic organization that maps the face onto central nervous system structures with remarkable fidelity. Fine discriminative touch from the face travels through the principal trigeminal nucleus in the pons, which corresponds functionally to the dorsal column nuclei in the medulla. Pain and temperature information from the face descends through the spinal trigeminal tract to synapse in the spinal trigeminal nucleus, which extends from the pons down into the upper cervical spinal cord and represents the head's equivalent of the dorsal horn pain processing circuits. Somatotopic organization represents a fundamental principle of somatosensory system design, creating orderly maps of the body surface within each processing station from the spinal cord to the cerebral cortex. These maps are not simple geometric representations but rather reflect the functional importance and innervation density of different body regions. Areas requiring precise sensory discrimination, such as the fingertips and lips, occupy disproportionately large representations compared to regions like the back or proximal limbs where spatial resolution is less critical. The primary somatosensory cortex in the postcentral gyrus contains the most detailed and well-studied body map in the nervous system, known as the sensory homunculus. This distorted representation of the human form reflects the relative cortical space devoted to processing information from different body regions, with the hands, face and lips receiving enormous cortical territories while the trunk and proximal limbs are represented by relatively small areas. Special sensory pathways follow distinct anatomical routes that reflect their specialized functions and evolutionary origins. The visual system begins with photoreceptors in the retina that convert light energy into electrical signals, which are then processed through multiple stages of retinal circuits before being transmitted to the brain through the optic nerves. The partial decussation at the optic chiasm ensures that each hemisphere receives visual information from the contralateral visual field, enabling binocular vision and depth perception. Visual information travels from the retina through the lateral geniculate nucleus of the thalamus to reach the primary visual cortex in the occipital lobe. This pathway maintains precise retinotopic maps that preserve the spatial relationships present in the visual world, allowing the brain to reconstruct detailed visual images from the patterns of neural activity generated by retinal photoreceptors. The auditory system processes sound waves through a series of anatomical relays that extract information about frequency, intensity, and spatial location. Sound waves are converted to electrical signals by hair cells in the cochlea, which maintain a precise tonotopic organization where different frequencies activate hair cells at different locations along the basilar membrane. This frequency mapping is preserved throughout the ascending auditory pathways, creating tonotopic maps in each processing station from the cochlear nuclei to the auditory cortex. Binaural processing for sound localization begins in the superior olive complex within the brainstem, where neurons compare the timing and intensity of sounds reaching the 2 ears. These computations allow the auditory system to determine the direction and distance of sound sources with remarkable precision, capabilities that are essential for navigation, communication, and survival in complex acoustic environments. Vestibular pathways carry information about head position and movement from the semicircular canals and otolith organs of the inner ear to brainstem nuclei that coordinate balance, eye movements, and postural responses. The vestibular system operates largely below the level of consciousness, continuously providing the nervous system with information about spatial orientation and movement that is integrated with visual and proprioceptive inputs to maintain equilibrium and spatial awareness. Processing principles common to all sensory systems include receptive field organization, lateral inhibition, and adaptation mechanisms that optimize information extraction and transmission. Receptive fields define the spatial or stimulus domains that influence individual neurons, with neurons at different processing levels having receptive fields of different sizes and complexity. Early processing stages typically have small, simple receptive fields that preserve detailed spatial information, while higher processing areas have larger, more complex receptive fields that integrate information across broader regions. Lateral inhibition enhances contrast and sharpens spatial boundaries by allowing strongly activated neurons to suppress the activity of their neighbors. This mechanism improves the detection of edges, transitions, and other features that are important for pattern recognition and stimulus discrimination. Adaptation allows sensory systems to adjust their sensitivity based on the average level of stimulation, preventing saturation during intense stimulation while maintaining sensitivity to stimulus changes. Gate control mechanisms in pain processing demonstrate how sensory pathways can be modulated by other inputs and by descending control systems from the brain. The interaction between large and small diameter primary efferents at the spinal cord level can either facilitate or inhibit pain transmission, explaining how mechanical stimulation such as rubbing an injured area can reduce pain perception. Motor pathways represent the neural networks that transform intentions and decisions into coordinated physical actions, translating the abstract concept of movement into the precise patterns of muscle activation required for everything from delicate finger movements to powerful athletic performances. These descending systems originate in various regions of the brain and converge upon the motor neurons of the brainstem and spinal cord, creating hierarchical control systems that enable both automatic postural adjustments and skilled voluntary movements. The upper motor neuron systems encompass all descending pathways that originate from the brain and influence movement either directly or indirectly through their effects on brainstem and spinal circuits. These systems provide the descending commands that initiate movement, coordinate complex motor patterns, and modulate the excitability of lower motor neurons based on behavioral goals and environmental demands. The corticospinal tract represents the most direct pathway for voluntary motor control, carrying commands from the cerebral cortex directly to spinal motor neurons and interneurons. This massive pathway contains approximately 1 million axons that originate primarily from the primary motor cortex and the precentral gyrus, though significant contributions also arise from the premotor cortex, supplementary motor area, and posterior parietal cortex. The diversity of cortical origins reflects the complex nature of voluntary movement, which requires integration of motor planning, sensory guidance, and spatial awareness. Within the primary motor cortex, different body regions are represented in an orderly topographic map known as the motor homunculus. This distorted representation of the human form allocates cortical space based on the complexity and precision of movement control required, with areas demanding fine motor control such as the hands and face receiving disproportionately large cortical territories. The lips, tongue, and fingers occupy enormous regions of motor cortex, reflecting the sophisticated neural control required for speech and manual dexterity that characterize human behavior. The corticospinal tract follows a predictable anatomical course from the motor cortex through the internal capsule, cerebral peduncles, and brainstem before reaching the spinal cord. At the pyramidal decussation in the medulla, approximately 90% of corticospinal fibers cross to the opposite side of the nervous system, forming the lateral corticospinal tract that provides precise control over limb and digit movements. The remaining 10% continue uncrossed as the anterior corticospinal tract, which primarily influences axial muscles involved in posture and bilateral movements. The corticoblubber tract parallels the corticospinal system but targets motor neurons in the brainstem that control the muscles of the head and neck. This pathway enables voluntary control over facial expressions, eye movements, chewing, swallowing, and speech production. Many corticoblubber connections are bilateral, providing some redundancy that can preserve function even when one hemisphere is damaged. though certain functions, such as fine motor control of the lower face, show predominantly contralateral innervation. Extrapyramidal motor systems include all descending pathways that do not travel through the medullary pyramids, encompassing the vestibulospinal, reticulospinal, rubrospinal, and tectospinal tracts. These systems evolved earlier than the corticospinal tract and provide essential functions related to postural control, balance, and automatic motor responses that operate largely below the level of consciousness. The vestibulospinal tracts originate from the vestibular nuclei in the brainstem and carry commands that adjust posture and limb position in response to changes in head position and movement detected by the inner ear. The lateral vestibulospinal tract facilitates extensor muscles throughout the body, providing the anti-gravity support necessary for upright posture, while the medial vestibulospinal tract influences neck and upper limb muscles to coordinate head and eye movements during locomotion and postural adjustments. Reticulospinal tracts arise from the reticular formation in the pons and medulla, providing descending influences that modulate muscle tone, posture, and automatic motor patterns such as locomotion and breathing. These pathways can either facilitate or inhibit motor neuron activity depending on their specific origins and targets, creating a flexible system for adjusting overall motor excitability based on behavioral state and environmental demands. The organization of lower motor neuron systems reflects the final common pathway principle, where all motor commands, regardless of their origin, must ultimately influence the alpha motor neurons, whose axons directly innervate skeletal muscles. These motor neurons, located in the ventral horns of the spinal cord and motor nuclei of the brainstem, represent the bottleneck through which all voluntary and involuntary motor commands must pass to produce movement. Motor units consist of an alpha motor neuron and all the muscle fibers it innervates, representing the basic functional units of motor control. The number of muscle fibers per motor unit varies dramatically depending on the precision of control required, with muscles demanding fine control, such as those moving the eyes or fingers, having motor units containing only a few muscle fibers, while postural muscles may have motor units containing hundreds or thousands of fibers. The recruitment of motor units follows the size principle, where smaller motor neurons with low activation thresholds are recruited before larger motor neurons during increasing levels of voluntary effort. This orderly recruitment pattern ensures smooth gradations of force production and optimizes the efficiency of muscle contraction by activating the most fatigue-resistant motor units first and recruiting more powerful but less sustainable units only when higher forces are required. 3 massive fiber bundles. The cerebellar peduncles connect the cerebellum to the brainstem and provide the pathways for information flow between the cerebellum and other brain regions. The superior cerebellar peduncle carries output from the deep cerebellar nuclei to the thalamus and brainstem motor centers, while the middle cerebellar peduncle brings input from the pontine nuclei which relay information from the cerebral cortex. The inferior cerebellar peduncle carries input from the spinal cord, vestibular system, and inferior olive, providing the cerebellum with continuous updates about body position, balance, and movement execution. Anatomically, the cerebellum divides into the central vermis and 2 lateral hemispheres, each serving somewhat different functional roles. The vermis primarily controls axial and proximal limb movements, contributing to posture, balance, and locomotion. The cerebellar hemispheres have expanded dramatically during primate evolution and play crucial roles in the planning and coordination of skilled voluntary movements, particularly those involving the distal portions of the limbs. The cerebellum functions as a sophisticated learning machine, constantly comparing intended movements with actual performance and adjusting motor programs accordingly. This process, known as motor learning, allows for the gradual refinement of movement patterns through practice and experience. When you first attempt to ride a bicycle or play a musical instrument, your initial efforts are clumsy and poorly coordinated. However, through repeated practice, the cerebellum gradually learns to predict the sensory consequences of motor commands and adjusts these commands to improve performance. The climbing fiber system plays a crucial role in cerebellar learning by signaling movement errors to Purkinje cells. When a movement fails to achieve its intended goal, climbing fibers from the inferior olive become active, triggering complex spikes in Purkinje cells and initiating plastic changes at parallel fiber synapses. Over time, these changes modify the influence of parallel fiber inputs on Purkinje cells, gradually improving the accuracy and coordination of motor output. Beyond its well-established roles in motor control, the cerebellum contributes to balance and equilibrium through its extensive connections with the vestibular system. The vestibulocerebellum, located in the flocculonolar lobe, receives direct input from the vestibular organs and processes information about head position and movement. This region plays essential roles in maintaining balance during standing and walking, compensating for perturbations that might otherwise cause falls, and coordinating eye movements with head movements to maintain stable vision during locomotion. Recent research has revealed that the cerebellum also participates in cognitive functions traditionally associated with the cerebral cortex. The lateral cerebellar hemispheres receive extensive input from association areas of the cerebral cortex and project back to these same regions through thalamic relays, forming closed loops that contribute to cognitive processing. These cerebellar cortical circuits appear to play roles in language processing, working memory, attention, and executive function, suggesting that the cerebellum applies its computational principles not only to motor control, but also to cognitive operations. The cerebellum operates as a sophisticated timing mechanism, providing precise temporal coordination for both motor and cognitive processes. Many cerebellar functions require exquisite timing, from the coordination of muscle contractions during reaching movements to the temporal sequencing of syllables during speech production. Damage to the cerebellum often results in timing deficits that affect not only movement coordination but also speech rhythm and cognitive processing speed. Error correction represents another fundamental cerebellar function, implemented through the complex interactions between cortical and nuclear circuits. The cerebellum continuously monitors ongoing movements and compares them with internal predictions of sensory feedback. When discrepancies arise between intended and actual performance, cerebellar circuits generate corrective signals that adjust motor commands in real time. This error correction process operates at multiple timescales, from rapid within-movement corrections to longer-term adaptations that improve future performance. The clinical consequences of cerebellar damage underscore the importance of this structure in coordinated movement and balance. Cerebellar lesions typically produce ataxia, a characteristic syndrome involving poorly coordinated movements, intention tremor, and difficulty with balance and gait. Patients with cerebellar damage often exhibit dysmetria, an inability to accurately gauge the distance and force required for movements, and dystiadokokinesia, difficulty performing rapid alternating movements. These symptoms highlight the cerebellum's essential roles in movement coordination and motor learning. The spinal cord extends from the foramen magnum at the base of the skull to the level of the first or second lumbar vertebra, forming the crucial communication highway between the brain and the peripheral nervous system. This cylindrical structure, approximately 18 inches long and 3/4 of an inch in diameter, carries millions of nerve fibers that transmit sensory information upward to the brain and motor commands downward to muscles and organs throughout the body. The spinal cord organization reflects a segmental pattern inherited from our evolutionary past, with 31 distinct segments corresponding roughly to the vertebral column above. These segments include 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal segment, each giving rise to a pair of spinal nerves that innervate specific regions of the body. This segmental arrangement creates dermatomal and myotomal patterns that allow clinicians to localize spinal injuries based on sensory and motor deficits. 3 protective membrane layers, collectively called the meninges, surround the spinal cord and provide both physical protection and physiological support. The outermost layer, the dura mater, forms a tough fibrous sac that extends from the skull to the second sacral vertebra. This durable membrane creates the epidural space between itself and the vertebral canal, a region commonly accessed for anesthetic procedures during childbirth and surgery. Beneath the dura lies the arachnoid mater, a delicate membrane that creates the subdural space, though this potential space normally contains only a thin film of fluid. The innermost layer, the pia mater, adheres close to the spinal cord surface and extends laterally as the denticulate ligaments, which anchor the cord within the dural sac and prevent excessive movement that could damage neural tissue. The subarachnoid space, located between the arachnoid and pia mater, contains cerebrospinal fluid that provides buoyancy and cushioning for the spinal cord. This space communicates freely with the brain's ventricular system, allowing cerebrospinal fluid to circulate around both the brain and spinal cord. The lumbosystem, an enlarged portion of the subarachnoid space below the cord's termination, provides a safe location for lumbar puncture procedures to sample cerebrospinal fluid or administer medications. 2 prominent enlargements mark regions where the spinal cord accommodates the massive nerve supplies required for the upper and lower limbs. The cervical enlargement extends from the 3rd cervical to the 2nd thoracic segment, and contains the motor neurons and sensory processing circuits that serve the arms and hands. The lumbar enlargement spans from the first lumbar to the third sacral segment and houses the neural machinery for leg and foot control. The spinal cord terminates as the cornus medullaris at approximately the level of the first lumbar vertebra in adults, though this level varies somewhat between individuals. Below this point, the chorda equina, literally meaning horse's tail, consists of the nerve roots from lumbar and sacral segments that continue downward within the dural sac to exit at their appropriate vertebral levels. This arrangement occurs because the vertebral column grows longer than the spinal cord during development, creating a mismatch between spinal cord segments and their corresponding vertebrae. Internal examination of the spinal cord reveals a characteristic butterfly or H-shaped region of gray matter surrounded by white matter tracts. The gray matter contains the cell bodies of neurons, including motor neurons, sensory processing neurons, and interneurons that integrate and relay information between different levels and sides of the spinal cord. The central canal, a small channel filled with cerebrospinal fluid, runs through the center of the gray matter and represents the continuation of the brain's ventricular system. The gray matter divides into distinct regions called horns, based on their anatomical position and functional organization. The dorsal horns receive and process sensory input from the periphery, containing neurons that respond to touch, pressure, temperature, and pain stimuli. Different layers within the dorsal horn specialize in processing different types of sensory information, with superficial layers primarily handling pain and temperature, while deeper layers process touch and proprioceptive information. The ventral horns contain the motor neurons whose axons exit the spinal cord as ventral roots and innervate skeletal muscles throughout the body. These motor neurons are organized in columns that correspond to specific muscle groups, with medial columns controlling axial muscles involved in posture and medial columns controlling the muscles of the limbs. The largest motor neurons, called alpha motor neurons, directly control muscle contraction, while smaller gamma motor neurons regulate the sensitivity of muscle spindles. Lateral horns, present only in the thoracic and upper lumbar segments, contain the cell bodies of preganglionic sympathetic neurons that control autonomic functions such as heart rate, blood pressure, and glandular secretions. These neurons send their axons to sympathetic ganglia located along the vertebral column, where they synapse with postganglionic neurons that innervate target organs throughout the body. The white matter surrounding the gray matter consists of ascending and descending fiber tracts that carry information between the spinal cord and brain. These tracts are organized into distinct pathways based on their origin, termination, and function, creating a sophisticated information superhighway that enables rapid communication between different levels of the nervous system. Ascending tracts carry sensory information from the spinal cord to the brain, with different pathways specialized for different types of sensory input. The dorsal column pathway transmits fine touch, vibration, and proprioceptive information from the body to the brain, with fibers ascending ipsilaterally before crossing in the medulla. The spinothalamic tract carries pain and temperature information, with fibers crossing shortly after entering the spinal cord and ascending to the thalamus in the contralateral white matter. Descending tracts bring motor commands and modulatory influences from the brain to spinal motor circuits. The corticospinal tract, the largest descending pathway, carries voluntary motor commands from the cerebral cortex directly to spinal motor neurons and interneurons. Most corticospinal fibers cross at the pyramidal decussation in the medulla and descend in the lateral white matter, while a smaller number remain uncrossed in the anterior white matter. Additional descending pathways include the vestibulospinal tract, which carry balance-related commands from the vestibular nuclei, the reticulospinal tracts, which convey postural and arousal-related influences from the brainstem, and the robrospinal tract, which contributes to motor control and coordination. These pathways work together to provide the complex patterns of motor output required for coordinated movement and postural control. Reflex arcs represent the spinal cord's capacity for independent information processing, allowing rapid responses to potentially harmful stimuli without requiring brain involvement. The simplest reflex arc involves only two neurons, a sensory neuron that detects a stimulus and a motor neuron that produces a response. However, most reflexes involve interneurons that can integrate information from multiple sources and coordinate complex response patterns. The stretch reflex exemplifies the elegance of spinal cord circuitry, involving muscle spindle receptors that detect muscle lengthening and monosynaptically excite the motor neurons that innervate the same muscle. This reflex helps maintain muscle tone and resist external forces that might disrupt posture or movement. Reciprocal inhibition, mediated by inhibitory interneurons, simultaneously relaxes the antagonist muscles, ensuring coordinated movement patterns. Propriospinal connections link different segments of the spinal cord, allowing coordination between distant body regions and integration of sensory information across multiple segments. These connections enable complex motor patterns such as locomotion, where the activity of legs must be coordinated with arm movements, and postural adjustments. Central pattern generators within the spinal cord can produce rhythmic motor patterns for locomotion, even in the absence of brain input, though brain circuits normally modulate and control these basic patterns. The relationship between spinal cord segments and vertebral levels creates important clinical considerations, as damage to specific vertebral regions affects predictable patterns of neurological function. Cervical injuries can affect arm and hand function as well as respiratory control, while thoracic injuries primarily impact trunk and leg function. The level of injury determines not only which body regions are affected, but also the potential for recovery, as the spinal cord has limited capacity for regeneration compared to peripheral nerves. The cranial nerves represent 12 pairs of specialized neural pathways that emerge directly from the brain and brainstem, serving as the primary communication channels between the central nervous system and the structures of the head, neck, and many organs throughout the body. These nerves, designated by Roman numerals from 1 through 12, carry sensory information from specialized receptors to the brain and transmit motor commands to muscles and glands, enabling the complex functions that define human experience and survival. The first cranial nerve, the olfactory nerve, provides our connection to the ancient world of chemical sensing that governs much of animal behavior. Unlike other cranial nerves, the olfactory nerve consists of numerous small nerve bundles that pass through the cribriform plate of the ethmoid bone, carrying aromatic molecules dissolved in nasal mucus to the olfactory bulb. This direct connection between the external environment and the brain bypasses the thalamic relay stations used by most other sensory systems, allowing smells to trigger immediate emotional and memory responses through connections to the limbic system. The second cranial nerve, the optic nerve, transmits the rich tapestry of visual information that dominates human sensory experience. Approximately 1 million axons from retinal ganglion cells converge at the optic disc and travel through the optic canal to reach the brain. At the optic chiasm, fibers from the nasal half of each retina cross to the opposite side, while temporal fibers remain uncrossed, creating the anatomical arrangement that allows binocular vision and depth perception. The optic nerve technically represents an extension of the central nervous system rather than a true peripheral nerve, as it develops embryologically as an outgrowth of the brain itself. The eighth cranial nerve, the vestibulocochlear nerve, actually consists of two distinct functional components that serve hearing and balance. The cochlear division carries auditory information from the spiral organ of Corti in the inner ear, where sound waves are converted into electrical signals that the brain interprets as music, speech, and environmental sounds. The vestibular division transmits information about head position and movement from the semicircular canals and otolith organs, providing the spatial orientation that allows us to maintain balance and coordinate eye movements with head movements. The purely motor cranial nerves control the precise movements of the eyes, head, and tongue that enable complex human behaviors. The third cranial nerve, the oculomotor nerve, innervates 4 of the 6 extraocular muscles that control eye movements, as well as the muscle that elevates the upper eyelid and the smooth muscles that constrict the pupil and adjust the lens for near vision. Damage to this nerve can result in a characteristic drooping eyelid, outward deviation of the eye, and inability to focus on near objects. The fourth cranial nerve, the trochlear nerve, has the distinction of being the smallest cranial nerve, innervating only the superior oblique muscle that rotates the eye downward and outward. This nerve also has the longest intracranial course, and is the only cranial nerve that decussates completely, crossing from one side of the brainstem to innervate muscles on the opposite side. The sixth cranial nerve, the abducens nerve, controls the lateral rectus muscle that moves the eye outward, and its long course through the skull makes it particularly vulnerable to injury from increased intracranial pressure. The 11th cranial nerve, the accessory nerve, consists of cranial and spinal components that work together to control head and neck movements. The spinal component arises from motor neurons in the upper cervical spinal cord and travels upward through the foramen magnum before exiting the skull to innervate the sternocleidomastoid and trapezius muscles. These muscles enable the powerful head-turning and shoulder-shrugging movements, essential for human communication and physical expression. The 12th cranial nerve, the hypoglossal nerve, provides motor innervation to most of the muscles of the tongue, enabling the precise movements required for articulate speech, efficient chewing, and safe swallowing. The tongue's remarkable dexterity, essential for human language, depends on the coordinated action of intrinsic muscles that change the tongue's shape and extrinsic muscles that change its position within the mouth. The mixed cranial nerves combine sensory and motor functions, creating integrated systems for complex behaviors. The 5th cranial nerve, the trigeminal nerve, represents the largest of the cranial nerves and divides into 3 major branches that provide sensory innervation to the entire face and motor control to the muscles of mastication. The ophthalmic division carries sensation from the forehead, upper eyelid, and nose. The maxillary division innervates the cheek, upper lip, and upper teeth. The mandibular division provides sensation to the lower face and jaw, while also controlling the powerful muscles that close the jaw during chewing. The trigeminal nerve's sensory functions include not only touch and pressure detection, but also the perception of facial pain and temperature. The trigeminal system processes some of the most intense pain sensations humans can experience, including dental pain and trigeminal neuralgia, a condition characterized by excruciating facial pain triggered by light touch. The motor division of the trigeminal nerve controls the temporalis, masseter, and tetragoid muscles that generate the tremendous forces required for effective chewing. The 7th cranial nerve, the facial nerve, orchestrates facial expression while also carrying taste sensation from the anterior 2/3 of the tongue and controlling salivary and tear production. The motor component of the facial nerve innervates the muscles of facial expression, enabling the complex emotional communications that characterize human social interaction. Damage to this nerve, as occurs in Bell's palsy, results in facial paralysis that affects not only voluntary expressions but also protective reflexes, such as blinking. The facial nerve's sensory functions include taste perception mediated by specialized receptors on the tongue that detect sweet, salty, sour, bitter, and umami sensations. These taste signals travel through the chorda tympani, a branch of the facial nerve that passes through the middle ear, highlighting the intimate and optical relationships between different sensory systems in the head and neck region. The 9th cranial nerve, the glossopharyngeal nerve, serves multiple functions related to swallowing, taste, and autonomic regulation. This nerve carries taste sensation from the posterior third of the tongue and general sensation from the throat and middle ear. Its motor component innervates the stylopharyngeus muscle involved in swallowing, while its parasympathetic fibers control salivary production from the parotid gland. The glossopharyngeal nerve also carries important sensory information from the carotid body and carotid sinus, specialized receptors that monitor blood oxygen levels and blood pressure. The 10th cranial nerve, the vagus nerve, extends the farthest of any cranial nerve, living up to its name, which means wandering in Latin. This nerve provides parasympathetic innervation to most thoracic and abdominal organs, slowing heart rate, stimulating digestive activity, and coordinating the complex reflexes involved in breathing and swallowing. The vagus nerve also carries sensory information from the throat, larynx, and visceral organs, providing the brain with continuous updates about the body's internal state. The clinical examination of cranial nerves provides a systematic approach to neurological assessment that can localize lesions within the brainstem and identify peripheral nerve injuries. Testing olfactory function involves presenting familiar odors to each nostril separately. Visual field testing and examination of pupillary responses assess optic nerve function and the visual pathways. Eye movements are evaluated by asking patients to follow moving targets, revealing deficits in the oculomotor, trochlear, or abducens nerves. Facial sensation and jaw strength evaluate trigeminal nerve function, while facial expressions and taste testing assess the facial nerve. Hearing tests and balance assessments examine vestibulocochlear nerve function. The glossopharyngeal and vagus nerves are tested through evaluation of swallowing, gag reflexes, and voice quality. Accessory nerve function is assessed by testing neck and shoulder muscle strength, while hypoglossal nerve function is evaluated through tongue movements and speech clarity. Understanding cranial nerve anatomy and function provides insight into numerous neurological conditions and their underlying mechanisms. Cranial nerve palsies can result from trauma, infections, tumors, or vascular disorders, each producing characteristic patterns of dysfunction that reflect the specific anatomical pathways involved. The systematic evaluation of cranial nerve function remains one of the most powerful tools in neurological diagnosis, revealing the intimate connections between anatomical structure and clinical function. The peripheral nervous system extends beyond the protective confines of the skull and vertebral column, forming an intricate network that connects the central nervous system to every region of the body. This vast communication system consists of all neural tissue outside the brain and spinal cord, including the spinal nerves, their branches, and the specialized nerve plexuses that redistribute and reorganize nerve fibers to serve the complex innervation patterns required by the human body. Each spinal nerve emerges from the spinal cord through the union of two distinct roots that carry fundamentally different types of information. The dorsal root contains the axons of sensory neurons, whose cell bodies reside in the dorsal root ganglion, a swelling located just outside the spinal cord, but within the intervertebral foramen. These sensory fibers carry information about touch, pressure, temperature, pain, and proprioception from their respective body segments toward the central nervous system for processing and integration. The ventral root contains the axons of motor neurons, whose cell bodies lie within the ventral horn of the spinal cord gray matter. These motor fibers carry commands from the central nervous system to skeletal muscles, enabling voluntary movement and reflexive responses. The separation of sensory and motor functions into distinct roots reflects a fundamental organizational principle of the nervous system and provides the anatomical basis for understanding various clinical conditions that affect sensory or motor function selectively. The dorsal root ganglia represent unique structures within the nervous system, containing the cell bodies of sensory neurons that would otherwise reside within the central nervous system. These ganglia house neurons of various sizes, from large neurons with heavily myelinated axons that carry proprioceptive and touch information at high velocities, to smaller neurons with lightly myelinated or unmyelinated axons that transmit pain and temperature signals more slowly. The positioning of these cell bodies outside the central nervous system may provide evolutionary advantages related to metabolic support and repair mechanisms. After the dorsal and ventral roots unite to form a spinal nerve, the nerve immediately divides into several branches called rami. The dorsal ramus innervates the deep muscles of the back and the skin overlying the vertebral column, maintaining the segmental organization that reflects our embryological development. The ventral ramus, much larger than the dorsal ramus, supplies the muscles and skin of the lateral and anterior body wall, as well as the limbs. In the thoracic region, the ventral rami maintain their segmental organization as intercostal nerves that run along the spaces between the ribs. These nerves provide motor innervation to the intercostal muscles, essential for breathing, and sensory innervation to the skin of the chest and abdomen. The orderly, segmental arrangement of thoracic nerves contrasts sharply with the complex reorganization that occurs in the cervical and lumbosacral regions. The cervical plexus, formed by the ventral rami of the upper four cervical nerves, innervates the muscles and skin of the neck and produces the phrenic nerve, one of the most important nerves in the human body. The phrenic nerve travels through the chest to innervate the diaphragm, the primary muscle of respiration. Damage to the cervical spinal cord above the level of phrenic nerve origin can result in respiratory paralysis, highlighting the critical importance of this neural pathway for survival. The brachial plexus represents one of the most complex reorganizations of nerve fibers in the human body, formed by the ventral rami of the lower four cervical nerves and the first thoracic nerve. This intricate network redistributes nerve fibers through a series of divisions and unions that ultimately produce the nerves supplying the entire upper limb. The plexus organization allows nerve fibers from multiple spinal segments to combine in various patterns, ensuring that individual muscles receive innervation from multiple spinal levels and providing some redundancy that can preserve function even if single nerve roots are damaged. Major nerves emerging from the brachial plexus include the musculocutaneous nerve, which innervates the flexor muscles of the arm and provides sensation to the lateral forearm. The radial nerve, the largest nerve from the brachial plexus, supplies the extensor muscles of the arm and forearm and provides sensation to the posterior arm, forearm, and hand. The median nerve innervates most of the flexor muscles of the forearm and the thenar muscles of the hand, while also providing sensation to the palmar surface of the thumb and adjacent fingers. The ulnar nerve supplies the remaining flexor muscles of the forearm and most of the intrinsic muscles of the hand, contributing to the fine motor control essential for human hand dexterity. The axillary nerve innervates the deltoid muscle and provides sensation to the shoulder region. Each of these nerves follows specific anatomical pathways that make them vulnerable to injury at characteristic locations, producing predictable patterns of motor and sensory loss. The lumbar plexus, formed by the ventral rami of the first four lumbar nerves, gives rise to nerves that innervate the muscles and skin of the anterior and medial thigh. The femoral nerve, the largest branch of the lumbar plexus, supplies the quadriceps muscle group essential for knee extension and walking, while also providing sensation to the anterior thigh and medial leg. The obturator nerve innervates the adductor muscles of the thigh and provides sensation to the medial thigh region. At this very moment, as you settle into whatever comfortable space surrounds you, approximately 86 billion neurons are firing in precise orchestration within your skull. Each neuron connects to thousands of others, creating a network so vast and intricate that if you could somehow stretch out all the neural pathways in your brain, they would span over 100,000 miles. This extraordinary biological supercomputer operates continuously, requiring no conscious effort from you, processing sensations, generating thoughts, and maintaining the delicate balance that keeps you alive. The nervous system represents perhaps the most remarkable achievement in biological engineering. Within the confines of your skull and spine lies a network more complex than any supercomputer ever built, processing millions of signals simultaneously, while you remain blissfully unaware of its ceaseless activity. Tonight, as you prepare for rest, this extraordinary system will continue its vigilant work, monitoring your breathing, adjusting your heart rate, and eventually weaving the tapestry of dreams that will carry you through the night. Understanding the nervous system is to glimpse the very essence of what makes us human. Every thought that crosses your mind, every memory you cherish, every sensation you experience flows through its intricate pathways, from the delicate dance of neurotransmitters across microscopic synaptic gaps to the coordinated firing of neural networks that span vast territories of brain tissue. This system orchestrates the symphony of consciousness itself. As we embark on this journey through the architecture of the nervous system, we will discover how electrical impulses become thoughts and emotions, and how this 3-pound universe nestled within your head creates your entire subjective experience of reality. I would love to know where you are listening from and what time it is in your part of the world right now. Whether you are settling in for the evening or finding a quiet moment in your day, I am grateful you have chosen to spend this time exploring the wonders of neural anatomy with me. Let us begin this gentle journey into the remarkable world of neural anatomy. The nervous system begins with its most fundamental building blocks, individual cells that have evolved into specialized communication networks. At the heart of this system lie neurons, the excitable cells that transform electrical and chemical signals into the rich tapestry of human experience. These remarkable structures represent one of biology's most elegant solutions to the challenge of rapid, long-distance communication within a living organism. A typical neuron consists of several distinct components, each serving crucial functions in the transmission of information. The cell body, or soma, houses the nucleus and most of the cellular machinery responsible for protein synthesis and metabolic activities. This central hub ranges from 4 to 100 micrometers in diameter, making it substantially larger than most other cells in the human body. Within the soma, specialized organelles work tirelessly to maintain cellular function. The endoplasmic reticulum, studded with ribosomes, produces the proteins essential for neurotransmitter synthesis and membrane maintenance. Mitochondria, often called the powerhouses of the cell, generate the enormous amounts of energy required for neural signaling. A single neuron may contain thousands of these cellular power plants, reflecting the intense metabolic demands of neural communication. Extending from the soma like branches of an intricate tree, dendrites form what neuroanatomists aptly call the dendritic tree. These branching projections serve as the primary receiving stations for incoming signals, their surfaces studded with receptors that detect neurotransmitters released by neighboring neurons. The complexity of dendritic branching patterns varies dramatically between different types of neurons. Some interneurons possess elaborate dendritic trees that can receive input from thousands of other neurons simultaneously, while others maintain simpler architectures suited to more specialized functions. The surface area provided by these branching structures is enormous, creating vast territories for synaptic contact and information integration. The axon represents the neuron's transmission cable, a thin projection that carries electrical impulses away from the cell body toward distant targets. While some axons measure only a few micrometers in length, others extend remarkable distances. The longest human neurons stretch from the base of the spine to the tips of the toes, covering more than a meter of distance. In giraffes, these same types of neurons can extend several meters, creating some of the longest single cells in the animal kingdom. The internal structure of axons contains specialized transport systems that move materials between the cell body and distant terminals. Fast axonal transport can move vesicles and organelles at speeds of several hundred millimeters per day, while slow transport carries structural proteins and enzymes at rates measured in millimeters per day. At its terminus, the axon branches into specialized structures called axon terminals, which form synapses with other neurons, muscle cells, or glands. These terminals contain concentrated collections of synaptic vesicles, membrane-bound packages filled with neurotransmitter molecules ready for release. The precise architecture of these terminals determines the strength and reliability of synaptic transmission, with some synapses capable of releasing neurotransmitter with remarkable precision and speed. The precise point where the axon emerges from the soma, known as the axon hillock, deserves particular attention. This region contains an exceptionally high concentration of voltage-gated sodium channels, making it the most electrically excitable part of the entire neuron. Here, incoming signals from dendrites are integrated, and if sufficient, trigger the all-or-nothing electrical event we call an action potential. The threshold for triggering an action potential represents a critical decision point in neural processing, determining whether accumulated synaptic inputs will be transmitted to downstream targets, surrounding and supporting neurons throughout the nervous system. Glial cells outnumber neurons by approximately 10 to 1, yet they often receive far less attention despite their essential roles. These supporting cells come in several varieties, each contributing unique functions to neural operation. Oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system wrap their cell membranes around axons, creating multiple layers of insulation called myelin sheaths. This biological insulation dramatically increases the speed of electrical transmission, allowing signals to travel at velocities exceeding 100 meters per second along the largest myelinated fibers, astrocytes. Star-shaped glial cells found throughout the brain and spinal cord perform numerous housekeeping functions that keep neurons operating optimally. They regulate the chemical environment around synapses, provide metabolic support to neurons, and help maintain the blood-brain barrier, that crucial selective filter that protects neural tissue from potentially harmful substances circulating in the bloodstream. Microglia serve as the nervous system's resident immune cells, constantly surveying for damage, infection, or cellular debris that requires cleanup. The blood-brain barrier itself represents one of the nervous system's most important protective mechanisms. This selective barrier, formed by tight junctions between the cells lining brain blood vessels, prevents most substances in the bloodstream from directly accessing neural tissue. Only essential nutrients like glucose and oxygen can readily cross this barrier, while toxins, pathogens, and most medications are effectively blocked. This protection comes at a cost, however, as it also prevents many potentially beneficial therapeutic compounds from reaching brain tissue. Neurons communicate through precisely timed electrical events called action potentials. At rest, a neuron maintains a voltage difference of approximately -70 millivolts across its cell membrane, with the interior more negatively charged than the exterior. This resting potential results from the uneven distribution of ions, particularly sodium and potassium, maintained by energy-consuming pumps embedded in the membrane. When sufficient stimulation occurs, voltage-gated sodium channels open rapidly, allowing positively charged sodium ions to rush into the cell. This influx reverses the membrane voltage, creating the action potential that propagates along the axon at speeds determined by the axon's diameter and degree of myelination. The diversity of neuronal types reflects the nervous system's functional complexity. Sensory neurons detect environmental stimuli and convert them into electrical signals, while motor neurons carry commands from the central nervous system to muscles and glands. Interneurons, which vastly outnumber both sensory and motor neurons, form the intricate processing networks that analyze information, store memories, and generate the patterns of activity underlying thought and consciousness. Some neurons release excitatory neurotransmitters that increase the likelihood of action potentials in their targets, while others release inhibitory transmitters that reduce neural activity. This balance between excitation and inhibition allows the nervous system to fine-tune its responses with remarkable precision. The brain represents the pinnacle of nervous system evolution, a 3-pound masterpiece of biological engineering that houses the essence of human consciousness. Enclosed within the protective vault of the skull, this remarkable organ consists of approximately 86 billion neurons interconnected by trillions of synapses, creating networks of staggering complexity that give rise to thought, emotion, memory, and perception. The most prominent features of the brain are the two cerebral hemispheres, massive structures that dominate the cranial cavity and account for roughly 85% of total brain weight. These hemispheres, while appearing nearly identical from the outside, exhibit subtle asymmetries that contribute to the lateralization of brain function. The left hemisphere typically dominates language processing, mathematical reasoning, and analytical thinking, while the right hemisphere excels in spatial processing, artistic abilities, and holistic pattern recognition. This division of labor, however, represents tendencies rather than absolute rules, as both hemispheres collaborate extensively through dense networks of connecting fibers. The surface of each hemisphere, known as the cerebral cortex, displays a characteristic pattern of ridges and valleys that maximizes the surface area available for neural processing. The raised ridges, called gyri, and the valleys between them, known as sulci, create a folded landscape that packs approximately 2.5 square feet of cortical surface into the confines of the skull. This folding pattern is not random, but follows consistent anatomical landmarks that divide each hemisphere into 4 distinct lobes. The frontal lobe, occupying the anterior portion of each hemisphere, houses the primary motor cortex and vast expanses of association cortex, responsible for executive functions, personality, and complex decision-making. The precentral gyrus within this lobe contains the primary motor cortex, where voluntary movements originate. Here, different body parts are represented in a distorted map called the motor homunculus, with areas requiring fine motor control, such as the hands and face, occupying disproportionately large regions of cortical real estate. Behind the central sulcus lies the parietal lobe, dominated by the primary somatosensory cortex in the postcentral gyrus. This region processes touch, pressure, temperature, and proprioceptive information from throughout the body. Like the motor cortex, the somatosensory cortex contains a topographic map of the body, with the most sensitive areas receiving the greatest cortical representation. The remainder of the parietal lobe integrates sensory information from multiple modalities, creating our unified perception of space and our body's position within it. The temporal lobe, located beneath the lateral sulcus on each side of the brain, contains the primary auditory cortex and critical structures for memory formation and language processing. The superior temporal gyrus houses the primary auditory cortex, where sound waves are transformed into meaningful auditory perceptions. Deeper within the temporal lobe lie the hippocampus and associated structures, essential for forming new memories and navigating through space. The occipital lobe, positioned at the posterior pole of each hemisphere, dedicates itself almost entirely to visual processing. The primary visual cortex, located along the calcarine sulcus, receives direct input from the eyes via the optic radiations. This region processes basic visual features such as edges, orientations, and movement, while surrounding visual association areas integrate these elements into the rich, detailed visual world we experience. Beneath the cortical surface lies white matter, composed of millions of myelinated axons that form the brain's internal communication highways. These fibers fall into 3 main categories: association fibers that connect different areas within the same hemisphere, commissural fibers that link corresponding regions between hemispheres, and projection fibers that carry information between the cerebral cortex and deeper brain structures or the spinal cord. The most prominent commissural structure is the corpus callosum, a massive bridge of white matter containing approximately 200 million axons that enable communication between the cerebral hemispheres. This structure ensures that information processed in one hemisphere can be shared with the other, allowing for integrated brain function. When the corpus callosum is damaged or surgically severed, as sometimes occurs in the treatment of severe epilepsy, patients may exhibit split brain phenomena, where each hemisphere appears to function somewhat independently. Deep within the cerebral hemispheres lie clusters of gray matter known collectively as the basal nuclei, or basal ganglia. These structures, including the caudate nucleus, putamen, globus pallidus, and associated nuclei, form circuits essential for motor control, habit formation, and reward processing. The basal ganglia receive extensive input from the cerebral cortex and send processed information back through the thalamus, creating loops that modulate voluntary movement and contribute to learning and motivation. At the core of the brain sits the thalamus, often described as the brain's relay station. This bilateral structure, roughly the size of a chicken egg, consists of multiple nuclei that process and direct sensory information to appropriate cortical areas. Nearly all sensory input, with the notable exception of olfaction, passes through specific thalamic nuclei before reaching the cerebral cortex. The thalamus also plays crucial roles in consciousness, sleep, and arousal, with damage to certain thalamic regions causing profound alterations in awareness. Beneath the thalamus lies the hypothalamus, a small but incredibly important structure that serves as the brain's primary link to the endocrine system. Despite weighing only 4 grams, the hypothalamus controls essential functions including body temperature regulation, hunger and thirst, circadian rhythms and emotional responses. Through its connections to the pituitary gland, the hypothalamus orchestrates the release of numerous hormones that regulate metabolism, growth, reproduction and stress responses. The limbic system encompasses a collection of structures that form the emotional and memory processing centers of the brain. This system includes the hippocampus, crucial for memory formation and spatial navigation, the amygdala, which processes fear and emotional significance, and the cingulate cortex, involved in attention and emotional processing. These structures work together to evaluate the emotional significance of experiences, form lasting memories, and guide behavior based on past experiences and current emotional states. The brain's internal cavities, known as ventricles, form a connected system filled with cerebrospinal fluid. The 2 lateral ventricles, located within each cerebral hemisphere, connect through the interventricular foramina to the 3rd ventricle, positioned between the thalamic nuclei. The 3rd ventricle communicates with the 4th ventricle, located between the brainstem and cerebellum, through the narrow cerebral aqueduct. This ventricular system produces, circulates, and reabsorbs cerebrospinal fluid, providing buoyancy, nutrition and waste removal for neural tissues. Cerebrospinal fluid is produced primarily by the choroid plexuses, specialized vascular structures located within each ventricle. These structures filter blood plasma and secrete approximately 500 milliliters of cerebrospinal fluid daily, though only about 150 milliliters circulates within the system at any given time. This clear, colorless fluid provides physical protection by cushioning the brain within the skull, maintains optimal ionic concentrations for neural function, and removes metabolic waste products from brain tissue. The circulation of cerebrospinal fluid follows a precise pathway from its production sites in the choroid plexuses through the ventricular system and into the subarachnoid space surrounding the brain and spinal cord. From there, the fluid is reabsorbed into the venous circulation through specialized structures called arachnoid granulations, maintaining a delicate balance between production and absorption that keeps intracranial pressure within normal limits. The brainstem represents the most ancient and essential portion of the human brain, a compact region that serves as the vital control center for life itself, measuring only about 3 inches in length. This remarkable structure contains the neural circuits responsible for maintaining consciousness, regulating breathing, controlling heart rate and coordinating the basic functions that sustain life from moment to moment. Without the brainstem's continuous vigilance, higher brain functions would be impossible, making this small region the foundation upon which all other neural activities depend. The most inferior portion of the brainstem, the medulla oblongata, extends from the foramen magnum at the base of the skull to the inferior border of the pons. This pyramidal-shaped structure houses the neural control centers that regulate the most fundamental physiological processes. Within its confines lie the cardiac centers, clusters of neurons that continuously monitor and adjust heart rate and the strength of cardiac contractions. These centers receive input from baroreceptors throughout the cardiovascular system, allowing them to respond rapidly to changes in blood pressure and ensure adequate circulation to vital organs. Adjacent to the cardiac centers, the respiratory centers orchestrate the complex process of breathing. The medullary rhythmicity center generates the basic respiratory rhythm, producing the cyclical patterns of inspiration and expiration that continue throughout life without conscious effort. This center coordinates with the pneumotaxic and apneustic centers in the pons to fine-tune breathing patterns based on metabolic demands, emotional states, and voluntary control from higher brain regions. The precise timing of these respiratory cycles requires exquisite coordination between multiple neural networks, ensuring that oxygen delivery and carbon dioxide removal remain optimal under varying conditions. The vasomotor center within the medulla controls the diameter of blood vessels throughout the body, providing moment-to-moment regulation of blood pressure and blood flow distribution. This center receives input from pressure sensors in major arteries and adjusts vascular tone accordingly, constricting vessels to raise blood pressure during stress or dilating them to reduce pressure when appropriate. The integration of cardiac, respiratory, and vasomotor functions within the medulla creates a unified system for maintaining cardiovascular homeostasis. Several cranial nerve nuclei reside within the medulla, serving as gateways between the central nervous system and the periphery. The hypoglossal nucleus controls tongue movements essential for speech and swallowing, while the dorsal motor nucleus of the vagus nerve regulates parasympathetic functions throughout the thorax and abdomen. The nucleus tractus solitarius receives sensory input from the facial, glossopharyngeal, and vagus nerves, processing taste information and visceral sensations that inform the brainstem's regulatory centers about the body's internal state. Perhaps the most anatomically striking feature of the medulla is the pyramidal decussation, where approximately 90% of the fibers in the corticospinal tracts cross from one side of the nervous system to the other. This crossing explains why damage to the left cerebral hemisphere typically produces motor deficits on the right side of the body, and vice versa. The remaining 10% of corticospinal fibers continue uncrossed as the anterior corticospinal tract, providing some degree of bilateral motor control. Superior to the medulla lies the pons, a bulbous structure that serves as a bridge connecting various regions of the central nervous system. The name pons literally means bridge in Latin, reflecting its role as a major crossroads for neural pathways traveling between the cerebrum, cerebellum, and spinal cord. The anterior surface of the pons displays prominent transverse fibers that carry information between the cerebral cortex and the cerebellar hemispheres, enabling the precise coordination of movement and balance. Within the pons reside the neural circuits responsible for generating and maintaining sleep, making this structure essential for consciousness regulation. The pontine sleep centers coordinate with the reticular formation and hypothalamus to orchestrate the complex transitions between wakefulness, non-REM sleep. During REM sleep, specific pontine nuclei become highly active, generating the rapid eye movements and vivid dreams characteristic of this sleep stage, while simultaneously inhibiting motor neurons to prevent the physical enactment of dream activities. The pons also houses the nuclei of several cranial nerves, including the trigeminal, abducens, facial, and vestibular cochlear nerves. The facial nerve nucleus controls the muscles of facial expression, allowing for the complex emotional communications that characterize human social interaction. The trigeminal nerve nuclei process sensory information from the face and control the powerful muscles of mastication. These cranial nerve functions require precise coordination with higher brain centers to produce appropriate responses to sensory input and emotional states. Respiratory control centers within the pons work closely with those in the medulla to refine breathing patterns. The pneumataxic center helps coordinate the transition between inspiration and expiration, preventing overinflation of the lungs and ensuring smooth respiratory rhythms. The apneustic center, when active, prolongs inspiration, though its function is normally modulated by input from the pneumataxic center and higher brain regions. The most superior portion of the brainstem, the midbrain, extends from the superior border of the pons to the posterior commissure. Despite its small size, the midbrain contains several crucial structures that integrate sensory and motor functions. The superior colliculi, located on the dorsal surface of the midbrain, process visual information and coordinate visual reflexes, such as tracking moving objects and directing gaze toward visual stimuli. These structures receive direct input from the retina as well as processed visual information from the occipital cortex, allowing them to integrate basic visual detection with complex visual analysis. The inferior colliculi serve as major relay stations in the auditory system, receiving input from multiple brainstem auditory nuclei and projecting to the thalamus. These structures play essential roles in sound localization, allowing organisms to determine the direction and distance of auditory stimuli. The precise timing of auditory inputs from both ears is analyzed within the inferior colliculi, creating the spatial auditory maps that guide orientation responses to sounds. Deep within the midbrain lies the substantia nigra, a darkly pigmented nucleus that produces the neurotransmitter dopamine. The neurons of the substantia nigra project to the basal ganglia, forming a crucial component of the motor control circuits. Degeneration of these dopamine-producing neurons leads to Parkinson's disease, highlighting the importance of this small brainstem structure in maintaining normal movement patterns. The red nucleus, another prominent midbrain structure, receives input from both the cerebral cortex and the cerebellum, integrating signals related to motor control and coordination. This nucleus gives rise to the robust spinal tract, which influences motor neurons controlling proximal muscles and contributes to the maintenance of posture and gross motor coordination. Surrounding the cerebral aqueduct within the midbrain, the periaqueductal gray contains neural circuits involved in pain modulation and defensive behaviors. This region can suppress pain perception during stressful situations, allowing organisms to continue functioning despite injury. The periaqueductal gray also coordinates behavioral responses to threat, including the fight-or-flight reactions that prepare the body for emergency action. Throughout the brainstem extends the reticular formation, a complex network of nuclei and interconnecting pathways that serves multiple essential functions. This system maintains consciousness and arousal, regulates muscle tone, and coordinates various autonomic functions. The reticular activating system within this formation determines which sensory inputs reach conscious awareness, filtering the constant stream of sensory information to prevent sensory overload while ensuring that important stimuli receive attention. The cerebellum, nestled beneath the occipital lobes and posterior to the brainstem, represents one of the nervous system's most architecturally distinctive and functionally sophisticated structures. Often called the little brain, this remarkable organ contains more neurons than all other brain regions combined, despite accounting for only 10% of total brain volume. Its surface displays a characteristic pattern of narrow, closely spaced folds called folia, which dramatically increase the available surface area and pack approximately 100 billion neurons into a relatively compact space. The cerebellar cortex exhibits a remarkably uniform three-layered organization that remains consistent across all regions of the structure. The outermost molecular layer contains relatively few cell bodies, but houses an extensive network of parallel fibers and climbing fibers that form synapses with the dendrites of Purkinje cells. This layer serves as the primary site of information integration within the cerebellar cortex, where inputs from multiple sources converge to influence Purkinje cell activity. The middle layer, known as the Purkinje cell layer, contains the large, distinctive neurons that serve as the sole output cells of the cerebellar cortex. These neurons possess elaborate dendritic trees that extend into the molecular layer, where they receive two distinct types of synaptic input. Climbing fibers, originating from the inferior olive in the medulla, form powerful excitatory synapses that can trigger complex spikes in Purkinje cells. Parallel fibers, the axons of granule cells, form weaker but more numerous synapses across the Purkinje cell dendrites, providing the primary excitatory drive to these output neurons. The deepest layer, the granule cell layer, contains the highest density of neurons found anywhere in the nervous system. These small but numerous cells receive input from mossy fibers, which originate from various brainstem nuclei and carry information about intended movements, sensory feedback, and motor context. The granule cells process this information and send their axons, the parallel fibers, into the molecular layer, where they influence Purkinje cell activity across wide regions of cerebellar cortex. Deep within the cerebellum lie the deep cerebellar nuclei, four pairs of structures that serve as the primary output centers for cerebellar function. These nuclei receive inhibitory input from Purkinje cells and excitatory input from the same mossy and climbing fiber systems that innervate the cortex. This arrangement creates a sophisticated computational circuit where the cerebellar cortex modulates the output of the deep nuclei, refining and adjusting motor commands based on ongoing sensory feedback and learned motor patterns. Gamma motor neurons provide a parallel control system that regulates the sensitivity of muscle spindle receptors, ensuring that these proprioceptive sensors remain responsive to muscle length changes even during active muscle contraction. The coordinated activation of alpha and gamma motor neurons, known as alpha-gamma coactivation, maintains the fidelity of proprioceptive feedback during movement and enables the nervous system to detect unexpected changes in muscle length that might indicate external perturbations or obstacles. The neuromuscular junction represents the critical interface where neural commands are converted into muscle contraction through chemical synaptic transmission. Motor neurons release acetylcholine at these specialized synapses, triggering muscle fiber depolarization and the cascade of intercellular events that produce force generation. The reliability and precision of neuromuscular transmission is essential for coordinated movement, and disruptions at these synapses can produce profound motor disabilities. Motor control hierarchies organize the nervous system's approach to movement generation into multiple levels of processing, each contributing different aspects of motor function. The spinal level provides the basic machinery for reflexes and simple motor patterns, including the stretch reflexes that maintain muscle tone and the flexion-withdrawal reflexes that protect against harmful stimuli. Central pattern generators within the spinal cord can produce rhythmic motor patterns, such as locomotion, even in the absence of descending input from the brain. Brainstem-level control centers coordinate postural adjustments, balance responses, and automatic motor patterns that support voluntary movements. The vestibular nuclei, reticular formation, and superior colliculus work together to maintain equilibrium, orient the body towards stimuli of interest, and provide the stable postural foundation upon which skilled movements can be superimposed. Cortical level control enables the planning and execution of skilled voluntary movements that characterize human motor behavior. The motor cortex, premotor areas, and supplementary motor areas work together to translate intentions into appropriate muscle activation patterns, incorporating sensory feedback and learned motor programs to achieve behavioral goals with efficiency and precision. Cerebellar influences on motor control operate through extensive connections with both cortical and brainstem motor centers, providing error correction, motor learning and timing functions that refine movement execution. The cerebellum receives copies of motor commands and compares intended movements with sensory feedback about actual performance, generating corrective signals that improve movement accuracy and contribute to the gradual improvement of motor skills through practice. Basal ganglia circuits contribute to motor control through their influences on cortical motor areas, providing selection mechanisms that facilitate desired movements while suppressing unwanted motor patterns. These circuits play crucial roles in habit formation, movement initiation, and the automatic execution of learned motor sequences, with dysfunction in basal ganglia circuits contributing to movement disorders such as Parkinson's disease and Huntington's disease. The integration of multiple motor control systems creates the flexible and adaptable movement capabilities that enable humans to perform an enormous range of motor tasks, from the gross motor patterns of locomotion to the fine motor skills required for writing, playing musical instruments, or performing surgery. Understanding these motor pathways provides insight into both normal motor function and the neurological conditions that can disrupt movement control. As consciousness fades and sleep approaches, the nervous system doesn't rest. It transforms. The very networks we have explored tonight shift into different modes of operation, revealing the remarkable adaptability of neural circuits that have sustained life for millions of years. Your brainstem continues its vigilant monitoring of vital functions, adjusting breath and heartbeat for the night ahead, while the reticular formation modulates your level of arousal, gently guiding you toward the restorative depths of sleep. The thalamus begins to filter sensory inputs more selectively, allowing only the most urgent signals to penetrate your descending awareness. Sleep itself becomes a fascinating neurological phenomenon, as slow waves sweep across your cortex in synchronized patterns that consolidate the day's experiences and clear metabolic debris from neural pathways. During REM sleep, specific circuits will activate in precise sequences, weaving dreams from memory fragments and emotional residues, while motor pathways become inhibited to prevent the physical enactment of dream movements. Even in sleep's deepest stages, your nervous system remains a guardian, ready to rouse you if circumstances demand, while orchestrating the restorative processes essential for tomorrow's alertness. The autonomic nervous system continues its essential work, maintaining the delicate balance between parasympathetic restoration and the minimal sympathetic tone necessary for survival. The nervous system you have journeyed through tonight represents millions of years of evolutionary refinement, from primitive neural networks to the sophisticated architecture that creates human consciousness. As you drift towards sleep, carried by these very neural mechanisms, rest knowing that you are cradled within one of the universe's most remarkable achievements, a living system that transforms electrical impulses into the rich tapestry of human experience. Sleep well, knowing your nervous system stands watch